What are enzymes? Enzymes are proteins that catalyze reactions that would normally take place, but very slowly. What this means is enzymes lower the activation energy required to push the reaction forward. There are many different types of enzymes that fall into 6 different classes.
Class 1 enzymes are oxidoreductases, which catalyze the transfer of electrons between molecules, otherwise known as redox reactions. So over here, A minus plus B yields A plus B minus. A minus acts as the reductant, which is oxidized to A, and B acts as the oxidant, which is reduced to B minus. Redox reactions require a coenzyme to act as an electron acceptor during reactions. So in this reaction, alcohol dehydrogenase acts as the enzyme that catalyzes the oxidation of ethanol to acetaldehyde. And in this reaction, NAD+ acts as the coenzyme that accepts the electrons transferred by ethanol.
Class 2 enzymes are collectively known as the transferase group, where they catalyze the reaction of the transfer of a molecule from one compound to another. In this example, we can see that compound AB plus molecule C results in molecule A plus compound BC, where we see that the molecule B gets transferred from A to C. That molecule B can be any groups that include methyl, phosphate, thiol groups, just to name a few. In this example, we can see that the conversion of riboflavin to flavin mononucleotide is catalyzed by the enzyme riboflavin kinase, where we see that the transfer of a phosphate group in ATP gets moved over to the hydroxyl group of the riboflavin. This results in the two molecules, ATP, with the phosphate group removed and being attached over to FMN, or flavin mononucleotide.
The third class of enzymes are the hydro. In this example, some substrate AB is split into its two separate parts, A and B, by adding a water to it. In this example, glycogen synthase, which is an enzyme in your body, is inactivated because there's a phosphorus group on one of its serines. But the enzyme glycogen synthase dephosphatase can add a water to it, which splits this phosphate off and lets it do its thing in the cell.
Class 4 lysases. Lysases are enzymes that catalyze cleavage bond of carbon-carbon bond, carbon-oxygen bond, and carbon-nitrogen bond. It dissociates molecules from molecules A into molecule B and C. Unlike hydrolysis and oxidoreductase, it proceeds without water, oxidation, and reductions. And because it proceeds without water, oxidation, and reductions, the product will have to generate a double bond or a ring structure. For example, 2-phosphoglycerate would dissipate into phosphenol, pyruvate, and water.
The fifth class of enzymes are isomerases, which catalyze the conversion of one molecule to another. The atoms stay the same, but the chemical bonds in the molecule rearrange. In this example, dihydroxyacetone phosphate, or DAP, is converted into its isomer glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase. The atoms in both molecules stay the same, but this keto group switches with the hydroxyl group at the terminal carbon, turning it into glyceraldehyde 3-phosphate.
Class 6 enzymes are ligases, which are responsible for ligating or joining two molecules together. The general form for this reaction is A plus B yields AB. So an example of a ligase is DNA ligase, which is responsible for joining together DNA. So here we can see a broken piece of DNA, which uses DNA ligase to join these together. This reaction is fueled from ATP, which then yields ADP, resulting in a fixed DNA strand.